The overall goal of this procedure is to automatically detect estrogen-responsive cells in live zebrafish larvae. This is accomplished by first breeding 5X-ERE-GFP adult fish to generate estrogen reporter zebrafish embryos. Next, the embryos are placed in individual wells of a 96-well plate. Then, estrogens are added to the plate of embryos. Finally, the plate is scanned for fluorescence using an epifluorescent microscope with a motorized stage. Ultimately, fluorescent microscopy is used to show tissue-specific activity of estrogen receptors.
So the main advantage of using this technique over other traditional techniques like high throughput or high content screening is that tissue-specific fluorescence in zebrafish larvae can be imaged using just a standard epifluorescent microscope equipped with a motorized stage. No specialized plate reader is necessary.
Caution: chemicals with BPA and estradiol are hazardous and can have deleterious effects on the human fetus. Handle endocrine disruptors with care and always use proper personal protective equipment. In this demonstration, zebrafish will be exposed to the estrogens 17-beta-estradiol, bisphenol A, and genistein in various doses beginning at 48 hours post-fertilization. After breeding zebrafish and collecting and decorionating eggs according to the text, on the day of treatment, dilute thawed stock chemical solutions